All right, welcome back for another deep dive. Today we're tackling a topic that's absolutely crucial in the world of medicine. We're talking about phase 3 clinical trials. Um, those really big studies that determine whether a new drug actually makes it to market or not.
Yeah, that's right. Phase 3 trials are kind of the main event in drug development. They're these large-scale studies, randomized controlled, really the gold standard for figuring out if a new treatment is safe and effective.
And for this deep dive, we've got a whole bunch of sources on everything from preclinical research all the way to the regulations and even some statistics. It's going to give us a really good understanding of what makes a phase 3 trial truly robust. We want to know what makes the results of these trials trustworthy.
Exactly. And today our goal is to unpack these design principles, the things that make a phase 3 trial really stand up to scrutiny. We're going to look at statistical rigor, how they define the endpoints, and why these choices are so important for getting reliable evidence.
Okay, so let's jump right in. One of the first things that comes to mind when we think about well-designed trials is how patients are divided up. Who gets the new treatment, who gets the standard treatment, or a placebo. And that brings us to randomization and control groups. They're kind of like the foundation of these trials.
Absolutely. Randomization, it's all about minimizing bias. Friedman, Furberg, and Demetz, they talk about this a lot in their work. So when we randomly assign patients to different groups, we try to make these groups as similar as possible at the start of the trial.
Right. So it's not just about things we can see like age or how severe their illness is. It's about all those other factors that we might not even know about that could affect the outcome.
Exactly. It's about leveling the playing field. And there are even these more complex adaptive randomization methods where the chance of being assigned to a particular group can actually change during the trial based on the data. That can be really helpful in some cases, but also makes the statistics more complicated.
Oh, wow. I see. So it's really important to get that balance right between being efficient and making sure the data is still solid. Now, what about control groups? Ellison, Laprinzi, and Kugler, they really emphasize control groups. Why are they so crucial?
Well, think of it like this. You need a point of comparison, right? Without a control group, it's like trying to judge a painting in a completely dark room. You have no idea if the colors are vibrant or dull, if the composition is good or bad. The control group gives us that reference point. We can see if the new treatment is truly better than the existing standard of care or even a placebo.
It makes sense. You need that benchmark to know if the new treatment is really making a difference.
Our sources also talk about different trial designs like parallel arm factorial crossover. How do these designs affect the robustness of a phase 3 trial? That's a great question. The design of the trial has a huge impact on how we analyze and understand the data. Bayer, Piantadosi, and Sen, they've done a lot of work on this. So in a parallel arm trial, you have different groups running simultaneously, like parallel tracks. Each group gets a different treatment throughout the study. It's kind of the most straightforward design.
I see. So that's the classic setup where you have one group getting the new drug, another group getting the standard treatment, and maybe even a third group getting a placebo.
Exactly. Then you have factorial designs, which are a bit more complex, but really interesting. In a factorial design, you can actually test multiple interventions or different doses of the same drug at the same time. So it's like running multiple trials within one trial. You can even see how different treatments might interact with each other.
Oh, wow. So you can get a lot more information from one study. But that must make the analysis pretty complicated.
It does. It does. And then there are crossover designs where participants actually get different treatments in sequence. So one person might get the new treatment first and then the standard treatment or vice versa. This can be helpful for reducing variability because each person acts as their own control, but you have to be careful about carryover effects where one treatment might influence the effects of the next one.
That's interesting. So the way you structure the trial itself is really important for getting reliable answers. And of course, we need to think about what we're actually measuring to determine if the treatment is working. Let's talk about endpoints.
Endpoints, they're absolutely crucial. They're the specific outcomes we're looking at to see if the treatment is having the desired effect. And Wang and Bakai, they stress that these endpoints need to be clinically relevant. They have to matter to patients. They need to reflect a real benefit like living longer or having a better quality of life.
So it's not just about measuring something for the sake of it. It has to translate to something meaningful for the people who are actually taking the drug.
Exactly. And on top of that, endpoints have to be measurable. We need a way to assess them in a reliable and objective way. And there are different types of endpoints.
Oh, right. Our sources mention continuous, binary, and time-to-event endpoints. Can you give us a quick breakdown?
Sure. So continuous endpoints are things you can measure on a scale like blood pressure or cholesterol levels, binary endpoints are events that either happen or don't, like whether someone has a heart attack or not. And time to event endpoints are things like overall survival or the time it takes for a disease to come back. They measure how long it takes for a specific event to occur.
So the choice of endpoint really depends on what you're studying and what you expect the treatment to do.
Yeah, exactly. You want to pick the endpoint that's most relevant to the disease and the potential benefits of the treatment. And then we have surrogate endpoints, which we've talked about before in our preclinical deep dive. They're like stand-ins for the real clinical benefit.
Right. They're those biomarkers or measurements that we hope will predict a real improvement in health.
Exactly. And surrogate endpoints can be really tempting because they might show changes earlier than the real clinical outcome. But the key thing is that the link between the surrogate endpoint and the actual clinical benefit needs to be rock solid. If that connection is weak, then a drug that looks good based on the surrogate might not actually help patients in the long run.
So while surrogate endpoints can be useful as supporting evidence, they shouldn't be the only basis for approval.
Right, you want to see a real impact on those direct, clinically meaningful endpoints. Okay, so let's move on to the statistical side of things. One of the most important aspects of a phase 3 trial is having enough participants. Why is sample size so critical?
Well, you need enough data to be confident in the results.
Right, exactly. Wang, Bakai, and Yin, they all talk about how crucial sample size is. If you don't have enough participants, there's a risk of getting a false negative. That means you might conclude that a treatment doesn't work when it actually does.
So it's about having enough power to detect a real difference if one exists.
Yeah. You don't want to miss a potentially effective treatment just because your study was too small. And there are ways to calculate the necessary sample size.
How did that work?
Well, you need to consider a few things. First, how big of an effect do you expect to see with the new treatment? Then there's the variability of the data. The more variability, the more participants you need. And then there's the level of statistical power you want. That's basically the probability of correctly finding a real effect if it's there.
So it's like balancing all these factors to find the sweet spot where you have enough data without making the trial unnecessarily huge and expensive.
Exactly. Julius and Yin, they go into detail about these sample size calculations. And there's also the concept of sample size re-estimation, which Gad talks about. That's where you might adjust the sample size during the trial based on the data you've already collected. So if it looks like you need more participants to get a clear answer, you can add them in.
It sounds like flexibility is important even in a phase 3 trial. Now, besides the statistics, the way the trial is conducted and the data is handled are also really important for the trial's robustness.
Oh, absolutely. We need really strong methodological standards and rock-solid data integrity. Wang and Mackay are very clear about this. Every trial needs a well-defined protocol that outlines every detail of how the study will be run. Things like how participants will be recruited, the exact procedures for giving the treatment, how data will be collected, everything.
So it's like a comprehensive instruction manual to ensure consistency across all the different sites and researchers involved.
Exactly. And of course, the data itself has to be top-notch. If the data is flawed or incomplete, the conclusions you draw from the trial are meaningless.
Makes sense. You can't build a house on a shaky foundation. Our sources also mentioned good laboratory practices, or GLP, and the importance of electronic signatures and record linking. How do those fit into the picture?
So GLP is outlined in CFR Title 21, Part 58, and it mainly governs those non-clinical lab studies that happen before a drug even gets to clinical trials. But the principles of GLP quality, integrity, traceability, those apply to every stage of drug development, including phase 3. Basically, you need to be sure that the data you're basing your clinical trial on is reliable.
Oh, so it's all connected. The quality of the early data influences the quality of the later data.
Exactly. And electronic signatures and record linking, they play a crucial role in ensuring that the electronic data from the trial is accurate, reliable, and can't be tampered with. You can track every change, every edit, every piece of data. That's outlined in CFR Title 21, Part 11. So it's all about creating an audit trail and preventing data manipulation.
So it's a whole system of checks and balances to maintain the integrity of the data. Now, even with all these safeguards in place, I imagine there can still be unexpected challenges when you're running a huge phase 3 trial.
Oh, definitely. Yin and Halaby talk about how even with the best planning, things can pop up that you didn't anticipate. Adaptive designs, which are more common in earlier phases, can sometimes be used in phase 3, but you have to be really careful because you don't want to compromise the integrity of the primary analysis.
So there's a balance between being flexible and sticking to the plan.
Yeah. And then there's the cost factor. Turner points out that phase 3 trials are incredibly expensive, so pharmaceutical companies need to weigh those costs against the potential benefits of the new treatment.
It's a huge investment of time and resources. So, to sum it all up, what are the key ingredients of a robust and reliable phase 3 trial?
I'd say the most important things are randomization and appropriate control groups to minimize bias and isolate the effect of the treatment. Then you need clinically meaningful endpoints that are measurable in a reliable way. And of course, you need enough participants to have the statistical power to detect a real effect if it exists. And finally, rigorous methodology and unwavering data integrity throughout the entire trial are absolutely essential.
Those are the pillars that support the whole structure. It's fascinating to see how all these different elements work together to generate the evidence that drives medical progress.
It is. It's like a puzzle where each piece is critical to the overall picture.
And ultimately, it's about getting those new treatments to the people who need them.
Absolutely.
Well, thanks for this deep dive. It's been incredibly insightful to explore the world of phase 3 trials and see the level of detail and care that goes into designing them.
My pleasure. It's always great to talk about the science behind drug development.
And to our listeners, we hope you found this deep dive informative and maybe even a little bit inspiring. It's a reminder that even in a world of complex science, there are clear principles and careful processes that guide the search for new and effective therapies. So until next time, keep asking those questions and keep diving deep.